everybody, today we're learning all about cotton. We're going to be looking at this unique crop and how the College of Agriculture is helping farmers produce a quality crop that is used all over the world. Hey everybody, we're here at the Old Rotation on the campus of Auburn University with Dr. Steve Brown. So Dr. Brown, tell us a little bit about yourself and your role at the university.
My title is, or my responsibilities involve all things cotton. I'm an extension cotton agronomist, and I know a little bit about everything, producing cotton and processing cotton and selling cotton and so forth. So I'm a resource person for those that do research. I'm a resource person for farmers, for ag retailers and ag distributors. I try to help them make the most of what they input on cotton and doing the best of producing cotton across the state of Alabama.
All right, so you're going to talk to us a little bit about cotton itself and what it looks like, how to grow it and all that. So will you show us around the field?
Sure.
All right.
This is a cotton plant, and cotton is unique in the sense of, unlike our other row crops that we produce, this is actually a perennial plant, which means it's a tropical plant, and if it didn't have cold weather, it would keep on going and keep on growing. In fact, I had a brother-in-law who was stationed in the Middle East, and he showed me a picture, and he had cotton that was about 15 or 20 feet tall in his driveway. So we've adopted this plant over the last 150, 200 years to grow as an annual. And of course, we're trying to produce fiber, lint, which produces garments and pillows and sheets and so forth. But there's also great value in the seed for livestock. It's a premium livestock feed, a lot of protein, and so again, it's probably limited mostly to feeding dairy animals. So this is what we're seeking to do. We have a plant that has a flower bud, and it's actually called a square, but there's nothing square about it. It's actually triangular. And this is the preliminary flower bud, and after about 3 weeks, this flower bud becomes a bloom like we see here. It's a white bloom, and this is the first day that bloom has opened, and that fertilizes itself and becomes what we ultimately are after is a cotton boll. And this is a cotton boll, and it grows it expands for about 3 weeks and then continues to grow, and the fibers expand and elongate, and they grow for about 7 or 8 weeks depending on weather. And then ultimately this will open into the white fluffy cotton that we want to harvest in most situations beginning in late September through hopefully Thanksgiving or thereabouts. But this is the ultimate goal, and so a cotton picker can grab that out, and there's the seed, and there's the fiber. I can feel the seed. You see the seed there, and there's the fiber on it. And this is we're growing it for the fiber, and fiber has all kind of characteristics by which it's measured. The first thing is color. If we get a lot of weathering on it, it diminishes the color. It also, we measure fiber length and strength and thickness, and there are certain parameters that are just right for the spinning mill process. And if they fit that perfect, and there are different types of spinning, but it becomes more efficient to making yarn, and then of course making the other things that we make from cotton. So, and there are a lot of pests that attack cotton, insect pests most notably. The boll weevil was a dominant pest that came up out of Mexico and moved across the U.S. and came to Alabama probably in about in 1915, 1918, 1920 in there, and it really took the possibility of production. It really hurt the production, and I'm from South Alabama, and of course Enterprise celebrates the boll weevil because it compelled them to look at alternatives to cotton, which is peanuts, and actually cotton and peanuts make a great rotation together. But so they're insect pests, there are diseases that get on cotton, there are soil-borne like microscopic worms, nematodes that are problematic. So lots of weed problems. Cotton is a slow-growing plant initially, and so providing weed control, effective weed control, is very important to maximize production, and then even to make sure we have a clean crop when we harvest it. So.
Okay. So do mostly the bugs or pests, they're going after the bulb? Are the
boll, B-O-L-L. Yes, okay. There are some things that do feed on the leaves. Spider mites, which are tiny mites, and they get on the underside of the leaves, and they can suck the plant down, and so sometimes we control them, but most of the pests are attacking what we're really trying to produce, the seed and the lint. In nature, you don't really have a vacuum. If you think you have a silver bullet, it won't last long because pests come in, and stink bugs are a key pest for us. And this year, plant bugs have been a major pest for us. So a lot of our crops, including cotton, are vulnerable to a lot of things. So we scout those. We walk the fields, and scouting could imply a lot of different things, but the most common numbers is people are going to be looking at the crop at all growth stages and looking to find damage. For example, they would actually crack open bolls and look for possible warts as a sign of stink bug feeding. When a stink bug pierces the side of the boll wall, stink bugs love to feed on the seed, and when it pierces the boll wall to get to the seed, it often introduces bacteria or fungi, and so you'll see rotted lint here and a loss of that particular boll. Now, a lot of things going on, a lot of the management inputs and decisions. My entomologist colleagues talk about farmers making so many hundreds of thousands of decisions across Alabama each year to deal with insect pests. So very dynamic situation in terms of management. And a farmer enters a crop year with a plan, but like a good football team, you got to make adjustments and do the right thing at the right time and alter things to make the most at the end of the season. So farmers are very vigilant in that regard, and cotton, compared to say corn and soybeans, is a very high input crop. A lot of things go in terms of fertilization, pest control, equipment, very specialized equipment. So a lot of opportunities for cost, and farmers, of course, trying to make the most of each of those inputs to maximize profits.
Hey everybody, we're here at the E.V. Smith Research Center, and I'm here with Dr. Jenny Kobernick. So Dr. Kobernick, tell us a little bit about yourself and where we are today.
Sure. My name is Dr. Jenny Kobernick. I am a cotton and soybean breeder in the Department of Crop, Soil, and Environmental Science. We are at the plant breeding unit of E.V. Smith. For those of you that don't know, E.V. Smith is really kind of an umbrella of 4 to 5 different research centers that we have. And this particular one, located in Tallahassee, is dedicated to plant breeding and variety testing.
All right, cool. Well, can you show us a little bit about what you do in
Sure.
cotton? All right. So tell me, what is plant breeding? What does that mean?
Okay, so plant breeding is essentially just improving the crop for mankind's benefit. And so another way to look at it would be I add value to the crop. So if there's a trait that a particular plant doesn't currently possess, and when I say plant, I'm talking more about a variety, I can cross that with another variety to try to incorporate all those traits together into a nice package for the farmer to grow. Another way to look at it is like you go to the grocery store, right? And you see all different types of apples, right?
Yeah.
Well, those apples are all different varieties. They're all different types of the same type of apple. And so you don't think about it as much, but we have that in cotton and in our other row crops as well. So essentially you just make a cross, and a cross just means, bringing male and female together, and the magic happens.
There you go.
Yeah, and so we're standing in the crossing block, and this is where it kind of starts. Cotton is naturally self-pollinating. It can be pollinated by bees. well, outcrossed by bees is the term. But this crossing block here is very intentional. So we've got 4 different lines planted, and so we're going to have 16 different combinations that we're gonna get. So cotton's very predictable. So the cotton flower starts to develop, and you get something like this that's called a candle. And this is what we use to emasculate. So essentially we take off the male parts of the plant in order to be able to give it pollen from another source. Okay. So in a flower, the pollen is considered the male part. So you bring in pollen from a different source.
Okay.
So in order to do this, we come in the afternoon and we emasculate. So emasculate means we just remove all the male parts. And so essentially all I'm doing here is taking off the petals, and then I'm removing all the male parts and leaving just the female. So the pistil is left. This is the part that receives the pollen. This is considered the female part. And so in the afternoon we come through, we put a little paper straw on it, and we tag it. But essentially that's our marker. So the next day we'll come in and we'll grab a white flower from a different line. Cotton is very predictable. The first day, the first day of anthesis, first day of where the pollen is viable, it's in the white it's a white flower. The second day it turns pink. So you'll notice some of the pink flowers. The pink flowers are a couple days old. And so if you notice, give me a thumb. So I always do it on my thumbnail, but you can. Do you see the little pollen granules shedding?
Kinda, yeah.
Yeah. So essentially, you just take this, you remove the straw, and you just put it on the top of the plant, the top of the pistil. The pollen will travel down through the tubes inside of it and fertilize the ovules at the bottom. And then once we've made the cross, we take the pink tag off and we put a pre-labeled marker on it.
So that you know what you...
So we know what the cross is. And so in a few days, it'll start to, the pistil falls off. Well, see, here's the pistil, it's aged, and it can actually just be popped off, but a bowl starts to form. And so what you get is the bowl, you keep the tag on it the entire length of time, and then it opens up and you harvest each individual bowl separately.
Yeah.
To keep them separate. So we do for each cross combination we do around, I think we're doing around 40 to guarantee that we have enough seed for that particular line to move on to the next generation. So the bowl these bowls that we're generating typically have around 25 seeds in them. Whereas soybean has 3. So it's a little bit easier to make crosses in cotton than soybeans or some other crop because the flowers are so big and you can see that the pollen, you can see the pollen granules. But we wear little aprons with all of our gear in it, and we typically come out in the afternoon, we emasculate them all, we tag them, and we record how many we have, and then we also count how many open flowers we have on the other row that's going to be the pollinator.
Oh, okay.
And we usually run a crew around 8 to 10 people to come out and not only make crosses, but do other field work as well.
Oh, okay. And that's just student workers that are in the college.
Yeah.
That's really cool. What a cool project.
So when you make this cross, the parents coming together basically forms what's called an F1 offspring. And they are all genetically alike, but the genes coming together.
Yeah.
And then the seeds coming off this, we actually either send to the winter nursery in Costa Rica, or we do it in the greenhouse on campus, and we grow the seeds over the winter because they're all the same. So I don't make really any selections, and I don't really take any data on it. And then after that, the seeds coming out of Costa Rica or the greenhouse, are considered F2s, and they're planted in a field trial somewhere else on the farm. From the F2s, they go into what's called single plant selections, and then they go through a battery of tests. So I guess I need to kind of distinguish this too. So this is breeding. This really isn't research, so to speak.
Okay.
So a research project entails sort of more of usually a start and a stop, where the breeding is continuous. I'm constantly putting new lines in, and we go through this series of tests, almost like kind of a machine, to see if we can get a viable product on the back end. For me, I focus primarily on disease resistance. I want to make sure that yield is maintained in no matter what area that we're looking at, because you can have a fantastic trait, but if you don't have yield, the farmers aren't going to grow it. So the other thing that I look for is seed composition. So one of the first things that the Cotton Commission was interested in was increasing seed, because when they go to sell their cotton at the gin, they could potentially get a premium for having larger cotton seed. So looking at ways to increase the size of the seed, but in order to do that, I have to also see what yield component I need to adjust in order to maintain that yield, because seed size is sort of like fiber quality. Once you breed for one thing, your yield's going to go down. So it's a very big balancing act in what you breed for in cotton, because these traits are all controlled by many genes. And so it's not just a quick fix. It starts with the cross, and this is where the DNA is basically getting scrambled, and then we look at essentially all the sister lines that come out of it to see which ones have the desired traits of the mom and dad.
Yeah, that's cool.
After we make the crosses over there, like I mentioned, it goes to the winter nursery or goes to the greenhouse, and then we run it through a bulk trial. In other words, we put all the seed together for that particular cross that we've made, and we plant it down at the very end. In this field is what's called an early generation test, and essentially it's just a replicated trial. We are going to grow it, test it for yield and the fiber quality traits of interest. And then what we do is basically select superior lines coming off that trial. So if you think about it in terms of genes, everything is segregating and everything's all together. And what we do is we look at what's called the bulk population. We look to see whether or not population A performs better than population B. And a population, what I'm referring to as a population or a family, is what's coming off the cross. So a particular cross becomes a population. So all the individuals together, we look at how well they perform. If they're hitting certain selection criteria, I know that if they're hitting that target, there's individuals that are going to be better because it's an average, right? So now in this particular setup, I selected, I believe there's 3 in here, so each row is a individual population. I selected 3. And then we're going to take 2 individual plants. So as the plant grows, we're going to harvest all the cotton off that one plant. Okay, and so then we're going to have 200 individuals within that population to try to find those individuals that test higher in those yield and fiber quality categories. So all of your varieties coming out of for cotton, soybean, things that are not like grown as a hybrid. So corn's different. They all are derived from a single plant. So you make the cross and then you pretty much lock in the genetics, and then from that point forward it's testing to make sure how it performs is consistent through each generation moving forward. So we'll take individual single plants, and then from a single plant we'll have all the seed off that one plant. We'll grow this. Actually, it's over there, but let's pretend we'll grow this single row. And so then we'll harvest the row, and then from there we plant it a couple more times, and then we start doing it in different locations. And as we go through the process of taking away the ones that aren't performing and keeping the top ones, we eventually get to where they go into a regional network trial, and then Then they're filtered into Henry's variety test.
So I am here at the Seed Technology Center on the campus of Auburn University, and I'm here with Henry Jordan. And so Henry, tell us a little bit about what you do and how you're involved with cotton.
Okay, so I run the variety testing program here at Auburn, and our main purpose is to support our stakeholders, which will be growers, breeders, and industry. We'll get cotton seed in from the breeders, we'll get cotton seed in from our industry participants. We'll take that seed, we'll package it into small packets, we'll take it out to the farms, and we'll plant it in small plot research. So once we have the plots in the field, we'll monitor it all year. The experiment station personnel help maintain, manage the trials for us during the year. At the end of the season, once everything gets harvested, we'll get our yield results back here. We'll work on putting that on the website for all of our stakeholders where it's available for everybody. We'll take the samples from our harvest, which will be seed cotton. We'll take it down the hall where we'll gin it, and then we'll determine the percent lint, which is the true number that everybody wants to know at the end of the year.
Well, can you show us a little bit of what you do?
Yep, I can do that.
Right.
All right, so over here we have packages of seed that come in. These come from our industry and our breeding participants. We'll take these envelopes and we'll package seed in each one of these envelopes. This goes out to our research centers, and we package those over here on our seed counters.
Each bag have a different amount of seed that it's gonna, or each?
Depending on our research center and whatever their plot length and row length and a row spacing, they'll have different seed counts per envelope.
Oh, okay.
For cotton this year, we ended up with 23 cotton trials across all the locations. So we have cotton at our Tennessee Valley location, which is Belmina. We have cotton in Prattville, at E.V. Smith, at Wiregrass, at Bruton, and in Fairhope.
Okay. And is it all doing the same research on it, just in different locations?
Yes, so all we're wanting to measure are differences by variety. So we're not looking at anything as far as fertilizer or seeding rate or any of the normal research that a lot of the other people do. We just want to treat everything exactly the same and see which variety performs best at each location.
Oh, okay.
This cotton came from a cotton picker, so if we look inside, you can feel it, you can feel the cotton seed inside of it. So this is what we would call seed cotton. This is straight from the plant, just like it would be picked. So what we'll do is for each plot that we harvest, we'll pull a sample. Sometimes it's this size, sometimes it's a little smaller. And we'll run it through this cotton gin. So this cotton gin is going to separate the seed from the lint. So the lint is the white fluffy part, and then you have the seed in the inside. So as we run it through this gin, the seed is going to drop out into this blue container in the front, and the lint is going to go into this big white box on the back where we'll collect it. I'll give you a quick demo of it, and let's do about that size, and then that way you can see how much lint we actually generate from this size of a sample. So we'll take it up here and we'll feed it in. We don't want to feed in too much at the time, but we'll feed in a little bit at the time. The trash and the seed will fall out the bottom, and eventually it's going to it's going to spin like that. So we can stop the machine. We'll come over here and open this back section up, and then here is our lint from our sample, and it's real soft and fluffy.
Yeah, that's a lot. That didn't. You had like a small handful.
Yep, it was just a little bit of seed cotton to quite a bit of lint.
That's cool.
This is a small research tabletop gin. The gins that they use for actual production cotton, I mean, that's a whole ginning facility. It's a big, huge building that's got a bunch of different gins in it.
And so you'll measure,
We'll run
study on the
We'll run probably, 3,500 plus samples through here a season. And so what we're looking for is we're going to measure the total sample weight to begin with, and then we'll measure how much lint we get off of it, and then we can calculate the percent lint. So we know how much of that sample was seed, how much of it was lint, and we can do the conversion to actually calculate how much lint we had for yield.
Oh, okay. And this is what the producer wants,
Yep.
the lint. Good.
That's where the money comes from when they
Okay.
sell their cotton that comes off of the lint.
Oh, okay, cool. What do you do with the seeds?
So for our purposes, the seeds, we don't use them, they just get destroyed. But for commercial purposes, the gin actually either saves the seed or sells the seed. Sometimes it goes into feed products and other stuff. All right, so the main purpose of anything is the end product. So what is the end product of variety testing? The end product is the data. So we want to get that data to our stakeholders. We want it to be very timely. So not only do we post it on the website, we send out it through an email distribution list where you can sign up and subscribe to updates. But before we do that, I have to go through and do a statistical analysis on the data. So if a grower has cotton in his field, he's not going to make 1,000 pounds of cotton completely all the way across the field. Some spots he may make 700 pounds, some spots he make 1,200 pounds. You know, there's going to be variation in the field. So through the scientific method and doing the research, we try to account for that variation in the data. So just because two varieties are numerically different, they may not be statistically different. We can't detect a difference between them using statistics, or either we can detect a difference through them using statistics. So that's part of what the data analysis is for. And then we take that data, we post it on the website for all the stakeholders to see. And we do a lot of different things with the data. So we have a variety selection platform that we have contracted with a third party to help manage. And it is partially funded through Extension, the College of Ag, my program, but it is also funded through all of the commodity groups. So check off dollars from the Alabama Cotton Commission go to help fund this so that the growers will have access to it and have a better way to look at the results.
Well, cotton is a major crop in most of the southeast states. There's 17 cotton producing states in the United States, and that's the lower half. And it is a major impact, not just cotton, but all the subsidies that help with cotton, the tractor dealerships, the chemical sales, the seed sales. There's a lot of families that make their living through agriculture, and cotton is a large part of it. Students can get involved in extension, they can get involved in working with any of the equipment suppliers, and a lot of the new things are drones are becoming a big part of that, and all the computer aspects, because all the equipment now has computers and computer monitors, and you know, all the new folks, all the young students are really involved in that, and I think that's something exciting for them to do.
Can you go more in depth about what students, the opportunities for students if they're not wanting to be an actual producer of cotton?
Right.
All those inputs?
Sure. Of course, you can be a cotton farmer, or cotton farmers, you can provide labor. Labor is a tremendous need in all agriculture, and cotton farming would be nothing short of that. We have suppliers that would include equipment dealers. We have suppliers that would sell crop protection chemicals, fertilizers, seed. We have crop insurance folks. We have others that would be actually cotton scouts and even consultants. They would be actively engaged in looking at the crop. And many of the ag retailers that sell crop protection chemicals and fertilizers and seed, they may have representatives that go into the fields and help farmers make management decisions. You'd have other areas at the processing cotton on the latter end, the cotton ginning, and all that goes with that. There's you know, research jobs, there's extension jobs. There are jobs in the national NRCS that would help, soil management, the water management, and the different farm programs that are delivered to producers. Those would be jobs that would certainly touch cotton as well.
There you go. So what opportunities are there for students to work, maybe what you're doing or even beyond the scope?
Honestly, I find it very hard to turn down a student.
Yeah.
Honestly, because I always love it when students come along and they're eager to learn more about plant breeding. In fact, I've had 2 undergrads approach me this year, and they're like, I know that I want to go into plant genetics, and I know that I want to go into plant breeding. And I'm like, okay, come on, like, we're going to find you something to do. We're going to give you like a little pet project to work on. So a lot of times when I have students like that, I pair them up with a grad student. So the grad student basically has like a little person to kind of show them the ropes of everything. And so like even on rainy days, like I've been known to send my crew to the library to look up research articles. And a lot of them, like I'd have 1 or 2 that came back and was like, I really enjoyed that, like getting to come through the literature and find out where it is and what it looks like. But in terms of student workers, like we go through cycles. So we'll start out, walking fields, hoeing weeds. We do crossing blocks. They test for, one of the things I do have to test for, talking about the traits, I do have to make sure that my cotton doesn't have traits in it. So that's one of the things I do have to do. So they'll go through and they'll do, it's kind of like it's basically a strip test. And the students will kind of take the lead on that and make sure that if the plant comes back and it's got traits in it, we pull the plant. So there's a lot of opportunities. There's a lot of different types of tasks. I do some molecular work as well, like screening for particular traits, like we've done that with reniform nematode resistance. But if a student comes on with me, like there's an expectation that they gotta be out in the field.
Yeah.
Because really to see the big picture and the full scope of things, you really gotta be in the field.